In this last lecture for Unit 1, we're going to be talking about the physical examination of urine. And I don't know if you remember from the first lecture of this unit, I mentioned that the process of the urinalysis or the procedure is kind of broken down into 3 stages. The first stage of the urinalysis is the physical examination. The second stage is the chemical examination, which we'll be talking about in Unit 2. And then the last stage of the process is the microscopic examination, which we'll be talking about in Unit 3. So the physical examination of urine includes examination of the urine's color, clarity, and specific gravity. Those results provide important preliminary information and allows for correlation with the other chemical and microscopic results. So normal urine color can range from almost colorless to black, with normal variations due to normal metabolic functions, physical activity, and ingested materials or pathologic conditions such as bleeding, liver disease, or infection. To determine the color and clarity of the urine, it should be examined under good lighting conditions. You want to look down through the container against a white background. Common descriptions of normal urine color include pale yellow, dark yellow, and amber. The color of the urine is caused by the normal urine pigment is called urochrome, which is a product of endogenous, which is internal metabolism that is produced at a constant rate under normal conditions. The amount produced is dependent on the body's metabolic state and can be increased in certain conditions like thyroid conditions and if the patient is fasting, as well as if the sample is allowed to sit at room temperature. It's an indication of the concentration of the sample, so you can get an idea of the patient's state of hydration. Some other pigments that can be produced in urine are uroerythrin, which is a pink pigment seen in specimens that have been refrigerated when amorphous urates precipitate, and urobilin, which is an oxidation product of the normal urine constituent urobilinogen, gives the urine an orange yellow to orange brown color to urine that is not fresh, so it would not produce foam. Some abnormal urine colors. There's a chart in your book that correlates urine colors to causes. Some common abnormal colors include dark yellow, amber, and orange, red, pink, and brown, brown to black, and blue to green. So with your dark yellow, amber, or orange urines, most times this is probably going to be normal, but it can also signify the presence of the abnormal pigment bilirubin, which will be detected during the chemical examination, but its presence can be suspected if a yellow foam appears when the specimen is shaken. A large amount of white foam when shaken indicates the presence of protein in a sample. A urine that contains bilirubin may also contain hepatitis, so we want to make sure that we're practicing our safe laboratory procedures, standard precautions. Photooxidation of bilirubin can cause a yellow-green color to the urine. It's caused by the presence of the pigment biliverdin. Yellow-orange can also be caused by a patient who is taking pyridium or azo, these are over-the-counter remedies for a UTI. This can cause interference with the chemical examination that uses tests based on color reactions. And then these specimens can also cause a yellow foam when shaken, which might be mistaken for bilirubin. Going back to the patients who are on pyridium or azo, the orange color that is produced from patients being on those medications is a very, very bright orange. So it's, to me, I've never seen one that I confused with the presence of bilirubin, which is usually more of kind of an amber color. The peridium produces just a super bright orange color, and the sample is usually a little bit thicker too. Okay, with red, pink, or brown urines, the most common cause is going to be blood, but may range from pink to brown depending on the amount of blood, the pH of the urine, and the length of contact with the urine to the blood. Pink to red urine can be due to red cells, hemoglobin, or myoglobin. Myoglobin is a byproduct of muscle breakdown, but all of those will produce a positive chemical test for blood. When red cells are the cause, the urine will be pink to red and cloudy, whereas when the cause is hemoglobin or myoglobin, the urine will be red and clear. Hemoglobinuria resulting from the in vivo breakdown of cells will result in red plasma, but myoglobinuria, which is the result of skeletal muscle breakdown, will not produce red plasma. Urine specimens containing porphyrins, which is a pigment, can also appear red, resulting from the oxidation of porphobilinogen, but these urines are most commonly referred to as having a port wine color, which is a very, like a very dark burgundy. Other non-pathogenic causes of red urine include medications, menstrual contamination, and ingestion of highly pigmented foods such as beets or blackberries. There's just a visual representation of differentiating between hemoglobinuria and myoglobinuria and hematuria, and a little bit of information on that port wine color caused by porphyrins. Okay, brown or black urines. Additional testing should be performed on urine specimens that turn brown or black on standing, but have negative chemical results for blood because they may contain melanin or homogenesic acid. Melanin is produced in excess when there's a malignant melanoma present. Homogenesic acid, which is a metabolite of phenylalanine, will cause a brown color in alkaline urine in people with the inborn error of metabolism called alkaptonuria. And we're going to get more into the inborn errors of metabolism in Unit 4. Certain medications can also cause brown or black urines. Blue, green, and purple. Pathogenic causes are limited to bacterial infections, specifically infections with the organism Pseudomonas species, and intestinal tract infections resulting in increased urinary indican. Indican is a substance formed by the decomposition of tryptophan in the intestines, and it's excreted in the urine. Ingestion of chlorets breath mints can cause blue or green urine. Certain medication can cause blue urine. Some IV medications can cause green. And a purple color can be seen in catheterized specimens that is caused by either indican or bacterial infection with the organisms Klebsiella or Providencia. Okay, moving on to clarity. Clarity refers to how transparent or turbid a specimen is. So normal terms that we would use to describe clarity are clear, hazy, cloudy, turbid, milky, opaque. The clarity should be determined in the same way that color would be. And the terminology that your lab uses could be somewhat lab specific, or it might depend on the computer system you have, with, you know, which options it gives you for reporting clarity. Okay, normal clarity, freshly voided urine is usually clear, but a white cloudiness can occur when amorphous phosphates have precipitated in the urine. Non-pathogenic turbidity. So squamous epithelial cells and mucus often cause turbidity, especially in samples from females. Turbidity can also be seen on specimens that are allowed to stand at room temperature or refrigerated, and that's due to either bacterial growth that is not representative of the actual specimen or of amorphous precipitates. Amorphous phosphates and carbonates produce a white precipitate in urine with an alkaline pH, whereas amorphous urates are going to produce a pink precipitate in acidic urines. Some other non-pathologic causes of turbidity could be semen, fecal contamination, radiographic contrast media, talcum powder, and vaginal creams. With pathologic turbidity, the most common causes are going to be red cells, white cells, and bacteria. Other causes are large amounts of non-squamous epithelial cells, yeast, abnormal crystals, lymph fluid, and lipids. And the amount of turbidity seen in the sample should correlate with the amount of material that you see during the microscopic exam. Clarity is one of the criteria used for determining if a microscopic examination should be done. Not all clear urine is normal, and not all cloudy specimens are going to flag for anything on the dipstick. So if the urine is cloudy, then you would do a microscopic even if the chemical analysis is normal. Specific gravity, which we've touched on this topic just a little bit so far. This is an indication of the concentrating ability of the kidneys, and it determines whether the specimen concentration is adequate to ensure the accuracy of the chemical tests performed. Specific gravity of urine is a measure of the dissolved chemicals in the specimen, and it's influenced not only by the number of particles but also the size of the particles. So because of this, there may be a need to correct the result for the presence of particles that are not normally found in urine, such as glucose and protein. Currently, the only method in use that would require correction is the refractometer. Chemical reagent strips and osmolality would not require correction. A little bit of terminology here. An isothenuric urine, this is the term used to describe a urine with a specific gravity of 1.010 because this is the same specific gravity as the plasma filtrate entering the glomerulus. Hyposthenuric is a specific gravity below 1.010, and hyperstheniuric is specific gravity above 1.010. Normal random urines can range from 1.002 to 1.035 depending on the patient's state of hydration. Specimens that measure below 1.002 are probably not urine. So that is another, we talked in a previous lecture about how if you question the validity of a sample, whether it is actually urine or not, you can, specific gravity is another one of those tests you can perform to help you determine that. Most specimens measure between 1.015 and 1.030. Specimens exhibiting consistent low readings may require further testing. So some methods for determining specific gravity. The methods are broken down into direct and indirect methods. So your direct methods are urinometer, osmolality, and harmonic oscillation densitometry, and your indirect methods are through refractometer and the chemical reagent strip. So the urinometer, which is a direct method, consists of a weighted float attached to a scale that has been calibrated in terms of urine-specific gravity. The weighted float displaces a volume of liquid equal to its weight and has been designed to sink to a level of 1.000 in distilled water. So the additional mass provided by the dissolved substances in the urine causes the float to displace a volume of urine smaller than that of distilled water. The level to which the urinometer sinks represents the specimen's mass or specific gravity. This method is less accurate than other methods currently available, and it's just more time consuming. It's a little more cumbersome, so it's not really recommended. Corrections have to be made for temperature and high density substances in this method also. So you would have to subtract 0.001 for every 3 degrees the urine is below the calibration temperature, and then you would have to add 0.003 for every gram of protein present in the specimen, and you would have to add 0.004 for every gram of glucose present in the specimen. So again, it's just not a very user-friendly method. The refractometer, which is an indirect method, determines the concentration of dissolved particles in a specimen by measuring the refractive index. The refractive index is defined as a comparison of the velocity of light in air with the velocity of light in a solution. The concentration of dissolved particles in a solution determines the velocity and angle at which light passes through a solution. The prism in the refractometer determines the angle that light is passing through the urine, and it converts the angle to a calibrated scale to determine a specific gravity. Some advantages of this method are that a very small sample volume is required, so 1 or 2 drops is all you need to use a refractometer, and temperature corrections are not necessary. Results are less affected by particle size, but you would still have to correct for glucose and protein, and corrections are the same as for the urinometer. So the method the procedure for this, for the refractometer, you would place, and I'm going to skip to this picture so you can see it while I'm talking about it. So you would place a drop of urine on the prism. So this is a little like flap or window or door that opens up here. So you open it up and then place your drop of urine on the prism. Focus the instrument on a good light source, and then you take your reading directly from the specific gravity scale as you're looking through the viewfinder. And then you would want to wipe off the prism and cover between each specimen. To calibrate, you can use distilled water, which should give you a reading of 1.000, and you can adjust that set screw to correct the reading if necessary. Also, 5% sodium chloride can be used. It should read 1.022 plus or minus 0.001, or you can use a 9% sucrose solution, which should read 1.034 plus or minus 0.001. Okay, next, harmonic oscillation densitometry, which is a direct method, is based on the principle that the frequency of a sound wave entering a solution will change in proportion to the density of the solution. This method was used in the IRIS automated urinalysis systems, which a lot of laboratories use the IRIS system, but the method has been replaced by reagent strip analysis in a lot of instances. Okay, osmolality, which is a direct method, is affected only by the number of particles in the solution, not the density of the particles. So this is a more representative determination of concentration ability. In this method, urea, sodium, and chloride all contribute equally to the measurement since molecular weight does not matter. Serum and urine are both tested for osmolality, and a result from each sample is usually done so that the results can be compared. The unit of measure in this methodology is the milliosmol, and we already mentioned that vapor pressure and freezing point osmometers are the most common. And then, of course, the chemical reagent strip, which is an indirect method. So addition of specific gravity testing to the chemical reagent strips eliminated the need for performing an additional separate procedure. The reagent strip reaction is based on the change in dissociation constant of a polyelectrolyte in an alkaline medium. So the higher the concentration of the urine, the more hydrogen ions are released, which then lowers the pH. So that pH change causes color changes on the reagent pad, and the color reaction corresponds to a specific gravity result. This method is also not affected by molecular weight of substances, and we'll get more into this in the next unit, and we'll discuss exactly what is measured and what the indicators in the strip are. So for now, just know this little bit of information that we've gone over. Okay, clinical correlations for specific gravity. The only ones that we will worry about on this slide are the abnormally high results. Specific gravity results greater than 1.035 could be due to the following. So it could be due to radiographic contrast dye. So if the patient has had a recent radiology procedure in which they had to use contrast dye, that could affect the results. dextran or other IV plasma expanders, which are used to boost plasma volume in order to increase circulation, such as in cases of shock. The patient's clinical course should be checked, but the MLT is not going to be the one to do this. It would have to be done by the nurse or the doctor. Odor, so this is not part of the routine urinalysis. It's not something that we would routinely report, but it is a noticeable physical property. It's said that fresh urine has, quote unquote, a faintly aromatic odor, and the longer it sits, the more odor, the more that odor will start to resemble ammonia, and that's due to the breakdown of urea in the urine. Causes of unusual odors in the urine can include bacterial infections, which cause a strong odor similar to the ammonia odor of old specimens, and diabetic ketones, which produce a sweet or fruity odor. Other metabolic disorders can produce significant odors, and we'll get into those in a later chapter when we talk about those specific disorders. Ingestion of certain foods or medications can also cause distinct but not clinically significant odors. And just kind of as a fun fact, studies have shown that although everyone who eats asparagus produces that very distinct odor in the urine, only those people who are genetically predisposed can smell that odor. Okay, and then lastly here, there in this chart should be in your book as well, but just some common causes of urine odor. So they've got the description over here, and some of these I just don't know where they get these descriptions from. But then the cause over here. So if, like I said, this is not something that we would routinely report out on the urinalysis, in the urinalysis results, but if it was something that could be related to one of these pathologic conditions, then it may be something that we want to either make a note of on the report or just call the doctor and let them know. So this is the final lecture in Unit 1.